Squalene Monooxygenase, abbreviated SqMO, is an enzyme from the oxidoreductase family. It catalyzes the formation of an epoxide across a double bond of a squalene molecule, a precursor of lanosterol, the molecule from which all sterols are derived.
Currently, we use SqMO as an antifungal agent to prevent the formation of lanosterol, and by consequence other sterols, such as ergosterol, which is a key component in fungal cell membranes. If you've ever used tough actin tenactin to treat your athlete's foot, then you've benefited from inhibitors of SqMO.
So why do we choose to study SqMO?
Well, inhibitors of SqMO are currently valued for their therapeutic properties, and current research indicates that inhibitors of SqMO could one day be used to complement or to replace statins in the reduction of cholesterol levels.
How does this relate to class? Both the inhibition of SqMO and the subsequent domino reaction resulting in lanosterol relates to our class in each of the following ways: asymmetric induction, conformational changes during inhibition in the secondary and tertiary structures, general acid-base chemistry, and rational drug design.
Squalene monooxygenase is bound to the endoplasmic reticulum of the cell in association with flavin adenine dinucleotide, FAD, a redox cofactor bound to the enzyme. NADPH is a coenzyme that acts as a reducing agent and transfers electrons to the FAD, giving it a negative charge. The reduced FAD is now ready to accept molecular oxygen, which is then protonated to form a flavin hydroperoxide. During this time, the squalene molecule also moves into the active site of the enzyme. The flavin hydroperoxide functions like a peroxy acid to epoxidize the nearby double bond, culminating in the formation of 2,3-oxidosqualene. You should note that the mechanism of epoxidation is a little bit different than the one we learned in organic chemistry, but this is because of the enzyme activity. Furthermore, also take note of the general acid-base activity facilitated by the protein. The second part of the mechanism is catalyzed by only one enzyme, oxidosqualene lanosterol cyclase, which opens the epoxide via protonation and helps situate the molecule in a conformation conducive to a series of intramolecular additions and transfers. Incredibly, this enzyme is responsible for the formation of 4 rings, 6 carbon-carbon bonds, and 7 chirality centers, a great display of biological specificity. Thus, inhibition of SqMO prompts a deficit of lanosterol, meaning that its derivatives, such as ergosterol, the yellow membrane lipids on the screen in front of you, are no longer available, resulting in the death of fungal cells via lysis. No more athlete's foot. This visual, in the sequence that follows, are protein ribbon representations of the squalene binding site in SqMO that we will use to show how inhibitors of SqMO might work. Terbinafine, a synthetic allylamine inhibitor, is the active ingredient in such antifungal drugs as tough actin tenactin. It is a non-competitive inhibitor with 2 mechanisms of inhibition. One is a terp-butyl group that projects out of the enzyme and prevents the binding of squalene with the FAD domain of SqMO. Additionally, terbinafine is a lipophilic molecule and so contains a naphthalene ring hinged so as to position itself in the small, very hydrophobic interior of the upper binding area. This causes a conformational change in the structure of the enzyme and also prevents binding. Thus, we can see generally how the terbinafine acts to inhibit SqMO, but we talked earlier about using inhibitors of SqMO to replace statins one day. How might this happen?
Researchers are currently investigating the inhibition of SqMO with tellurium-containing compounds. These compounds are similar to terbinafine in that they are non-competitive inhibitors as indicated by this Lin-Weaver-Burke plot. They function as inhibitors by interfering with cysteine residues and thus disrupt the disulfide bonds that contribute to the enzyme structure and thus function. Additionally, they have been found to disrupt the general acid-base chemistry associated with both histidine and arginine residues in the enzyme.
This is a generalized pathway for the synthesis of cholesterol. We've highlighted 2 important enzymes. We currently use inhibitors of the enzyme HMGR, called statins, as drugs for the reduction of cholesterol levels. Researchers are now proposing that we instead use inhibitors of SqMO to realize the same goal. To see why this suggestion is in accordance with the principles of rational drug design, let's go back to HMGR. You can see that inhibition of HMGR also means that the products of its enzyme activity are no longer produced. This is notable because one of its products, coenzyme Q, is part of the electron transport chain vital to cellular respiration. In using inhibitors of SqMO, this product is still produced, and cells can continue to operate in more natural conditions.
Despite the potency of tellurium and terbinafine as inhibitors of SqMO, they are not practical in an internal physiological setting because they are toxic. That being said, the research that has been done on them provides valuable insight into the crucial domains of SqMO that could be targeted in the future.
At this point, we've come full circle, demonstrating concepts from class including asymmetric induction, conformational changes, and general acid-base chemistry. More importantly, we've used the principles of rational drug design to show that inhibitors of SqMO might be a bit better than statins in reducing cholesterol levels. Squalene Monooxygenase is certainly an enzyme worth keeping your eye on.